Hello, I'm Joe Takahashi from UT Southwestern and the Howard Hughes Medical Institute. And in this third lecture, I want to introduce you to the biochemistry of clock and BMAL and focus on the functions of those two important clock proteins. As we saw in the previous two lectures, the clock is controlled by a network of interacting genes with a primary negative feedback loop involving clock BMAL and the peripheral genes. And for this lecture today, what I want to do is really focus on what we call the core feedback loop, and in particular focus on clock and BMAL, the transcriptional activators in this system. And so a number of years ago, we began to focus on the biochemistry of these two clock proteins, clock and BMAL, which are illustrated here in diagrammatic form. And to study them, we attempted to express and purify those proteins. And we were able to express truncated forms of both clock and BMAL to high levels that were able to produce crystals, such as these shown here. And using this method, we are able to determine the X-ray crystal structure of the clock-BMAL complex, which is shown here on the right. BMAL in blue and clock in green. Interestingly, what we can see are the three major domains, the BHLH, or basic helix-loop-helix domain, the PAS-A domain, and then the PAS-B domain of BMAL in this particular region. And then here, the interaction of the two proteins. Interestingly, what we find in the crystal structure is that clock is actually wrapped around BMAL, and the interactions of the PAS domains are rather interesting and complex. The next slide just shows you a 3D view of the two proteins. And we'll just, in the next couple of slides, take a closer look at the PAS-A domain followed by the PAS-B domain. So this is the PAS-A domains of BMAL in blue and clock in green, looking at the domains down the long axis of the protein. And what you can see is that there's a helical, a helix that's N-terminal to the PAS domain, which is the rest of the structure here. And these two N-terminal helices form very interesting interaction interfaces between the complementary PAS-A domains, such that the alpha helix of one PAS-A domain interacts with the beta sheet surface of the other PAS domain. This kind of interaction has been seen in bacteria, in this case in the NIF-L protein. But this is really the first view of the PAS-A domain in the mammalian protein. Even more interesting, however, are the PAS-B domains of clock and BMAL, which are shown here, in which BMAL actually stacks in a parallel fashion with clock and BMAL, such that the alpha helical surface of clock is actually interacting with the beta sheet surface of BMAL. This is a little unusual because traditionally all other PAS-PAS domain interactions typically involve the beta sheet surfaces of those two subunits. As you can see in this example here for the HIF2 alpha PAS-B interacting with the ARNT PAS-B domain here, where the beta sheet surfaces of the two proteins are really the interaction interfaces. The next slide shows you a detailed view of this interaction, and what you can see is a very interesting tryptophan residue that is part of what's called the HI loop of BMAL. And in this surface view, you can see that this tryptophan residue inserts into a hydrophobic pocket on the surface of clock to form a very nice kind of lock and key interaction. And interestingly, this tryptophan residue is conserved not only in BMAL but in clock as well as the per proteins, which also have PAS domains. So this is just an overall summary of clock BMAL showing the location of a number of previously identified mutations in these two proteins. We can now place them on the structure and have some better understanding of how they might work. In particular, 5 mutations that interfere with cryptochrome binding all map to the HI loop of clock, including that tryptophan residue on clock. And so we see this as just really the first step in an atomic level understanding of how clock and BMAL might function. And in future work, we hope to be able to see how the cryptochrome protein might interact with the clock BMAL complex, as well as other components, such as the period proteins. So for the rest of this talk, what I'd like to do is to focus in on this aspect of the mechanism, and that is the genomic targets of clock. And using next generation sequencing technology, we can interrogate the genome for the binding sites of almost any transcription factor that we can study today using a method called chromatin immunoprecipitation followed by next generation sequencing. So chromatin immunoprecipitation, also called CHIP, is a way that we can precipitate using antibodies to DNA binding proteins, specific regions of DNA, which we then can purify, make into libraries, and then sequence. Then we map those sequences back to determine where those proteins were binding in the genome. So this is a browser view of a single known target gene of clock called DBP, shown here. And in the DBP gene, we can see 3 regions where BMAL, in this case, bind to the promoter, the first intron, and the second intron of BMAL. And each of these profiles, shown in blue, are ChIP-seq profiles for BMAL DNA binding taken at different times of day, 6 different times, 0, 4, 8, 12, 16, and 20. And what you can see is the amount of binding is dynamic and changes through the day. It's high between 0 and 12, and it's low at 16 and 20. And then this final lane here, labeled KO, is a knockout mouse control for BMAL, which is a control for antibody specificity. There should not be any binding, as you can see. So that's just one factor at one gene. Here's the same gene, DBP, but now we're looking at the DNA binding of all the major transcriptional regulators in the core feedback loop. BMAL, clock, NPAS2, which is the paralog of clock, PER1, PER2, CRY1, and CRY2. And what you can see is that the 3 activators all bind the same sites, and they have a very similar temporal pattern. They bind primarily from 0 to 12, which is the daytime. And then the repressors, PER1, PER2, and CRY2, also bind the same 3 sites, but their temporal pattern is different. They are binding between 12 and 20, which is the night. And then, interestingly, CRY1 shows a third pattern where it comes on to bind late at 20, peaks at 0, and then starts falling off at 4. So that's just one gene. What about a genome-wide view? So this is a representation of all the binding sites for each of the 6 major clock proteins across the entire genome in Heatmap format. So in the case of BMAL, shown way over here, there are about 5,900 sites in the genome, and the red shows the intensity of binding. And what you can see is that binding is highest between 0 and 12 with a peak around 8 in the case of BMAL. Similar pattern for clock with a peak at 8. And then the repressors PER1, PER2, and CRY2 are all binding between 12 and 20 across the genome. And then CRY1 has a different pattern, which turns out to be a bimodal pattern. It has both a 12-hour and a 24-hour binding pattern. But the primary peak is actually at 0 right here. The other feature, which is a little interesting, is that the cryptochrome proteins have many thousands of sites. So in the case of CRY1, there are 16,000 sites in the genome, many more than the 5,900 BMAL1 sites. And the PER genes also have, or the PER proteins also have many binding sites. In the case of PER2, over 7,000 sites. So where are these sites, and are they binding to similar sites in the genome? So this complicated Venn diagram, which is called a Chow-Ruski diagram, shows the 6-way overlap of clock, BMAL, PER1, PER2, CRY1, and CRY2. In the middle is 1,400 sites where all 6 factors bind the same site, 6 out of 6. And then around the perimeter are all the various combinations, 5 out of 6, 4 out of 6, 3 out of 6, 2 out of 6. And so you can see that that binding pattern is a little bit complicated. But interestingly, the other feature of this graph is that we find many sites for CRY1 and CRY2 and also PER2 that are separate. They are completely separate from clock BMAL, which is interesting because we thought previously that PER and CRY primarily interacted with clock BMAL. But this analysis is actually consistent with work from 2 other laboratories that has shown that cryptochrome and PER can interact with another class of transcription factor protein called nuclear receptors, which are very pervasive across the genome. So that was DNA binding occupancy. That doesn't tell us about function. And so to try to assess function, we used RNA expression across the genome. And in this case, what we did was to use whole transcriptome RNA sequencing. And so whole transcriptome means that we're sequencing total RNA that's just been depleted for ribosomal RNA. And this allows to see both messenger RNA as well as intronic RNA sequences. And so this is a genome browser view of the PER2 gene. The PER2 gene is on the opposite strand, so it's going this way. And in this case, we're taking time samples every 4 hours over a 48-hour period. Okay? And so the black histograms pointing down show the PER2 transcripts on this opposite strand. And you can see that they cycle. They're low at 0 to 4, then they peak around 12 to 16 here. Then they go down again. They're low at 24, 28, and then they peak again between 36 and 40. Because this sequencing is strand-specific, we found a very interesting result shown in red. These are reads on the opposite strand. They reveal an antisense transcript within the PER2 gene. And this transcript is also cycling, except it's high when PER is low. And that's shown in this graph here, where the sense transcript is shown in black and the antisense transcript is shown in red. So there's a antisense, antiphase transcript in per, which we don't really know the function of at this point. The other feature in this kind of sequencing experiment is that we can annotate the sequence reads in different ways. We can look at the reads that are only in introns, or the reads that are only in exons, or in the entire gene body. And so if we look at just the intron reads shown in blue in this upper graph, we see that they're cycling for per2, as well as the exon reads shown in red. They're both in phase, but the exon reads, of course, are higher. So in this case, for the per2 gene, both the intron and the exon reads are cycling. Why would we want to do this? Well, the intron reads are a representation of pre-messenger RNA. And so they allow us to estimate nascent transcription in this case. So if we then look across the entire genome, we can ask how many genes show cycling RNAs in the liver. And these heat map representations show you two answers to that question. The first is the number of genes with intron cycling RNAs. And then the second are the number of genes with exon cycling RNAs to represent messenger RNA. And what we find is in the case of intron genes, or cycling genes, there are about 1,400 that are cycling in the liver. And in the case of exon cycling genes, there are about 2,000. Okay? Now, the exon cycling genes are distributed in time throughout the day. This is a phase plot of the peak of those rhythms. And you can see that the peaks are really spread out to many phases. And this is what we saw over a decade ago with microarray experiments, which of course are interrogating mainly exon RNA sequences. But interestingly, when we look at the intron reads, we find a very interesting result, and that is that there is a coordinated peak of intronic RNA rhythms that peaks at CT15. That's about 3 hours after the beginning of the night for the mouse. And this coordinated transcription is very interesting. The other feature is that the intron cycling genes and the exon cycling genes do not largely overlap. There are only about 450 that have both intron and exon cycling transcripts that are detectable. Okay, so because of this burst of transcription that's happening at CT15, we're very interested in trying to understand what might be regulating this. And so we wanted to then look at the early steps in transcription by RNA polymerase II, shown up here. And this shows a typical sequence of events in the early stages of transcription, where first activators such as clock and BMAL bind to regulatory sequences and genes. They then recruit co-activators such as P300 and CBP to open up the chromatin and allow the formation of the pre-initiation complex by RNA polymerase II. In this case, we can detect this hypophosphorylated Pol2 with this particular antibody 8WG16. Then one of the first steps in initiation of Pol2 is serine 5 phosphorylation of the C-terminal domain. And again, we can detect this form of polymerase with a specific antibody for Pol2 serine 5 phosphorylation. So this is a browser view of the PER1 gene showing BMAL, PER2, P300, 8WG or Pol2, then the serine 5 Pol2, and CBP. And the first surprise is that Pol2 occupancy across the entire genome of the liver is highly circadian. So you can see that here at the PER1 locus, where there's a very strong peak at CT12, which corresponds with the RNA transcription. But if we look genome-wide at over 7,000 sites in the liver, genome-wide, Pol2 is being recruited to DNA in a circadian manner. This is really quite surprising and quite interesting. And if we look at the timing of the Pol2 signal, it occurs at CT 14.5, which is just a half an hour before that intron RNA peak that we just looked at. So this suggests that perhaps this burst of transcription is really a reflection of new transcription by Pol2 across the entire genome. Now to look at the coactivators P300 CBP, these show you some heat map representations of those. P300 is recruited by clock, and we see that P300 occupancy is actually high in the morning, CT6 or 5. CBP, on the other hand, has a very unusual pattern. It's bimodal. There's a bump in the morning and then a very strong peak at night. This is very surprising because CBP is an activator. It's recruited by BMAL1, yet there's a peak at night when BMAL1 is typically not there. Okay? So what might that mean? And then the second surprise is when we looked at the serine 5 initiated form of Pol2, shown here, it is also highly circadian across the entire genome, but the phase is different. It peaks at 0. And that phase is actually very similar to the phase of binding for Cry1. And so we looked at the overlap of serine 5, Pol2, and Cry1, and we see that there is significant overlap. I'll come back to say what we think that might mean. And in the case of CBP, we found that it interacts with the repressor PER2, which is also very surprising since CBP is typically a coactivator and PER2 is a repressor. And so in the case of CBP, we think that perhaps in the morning it interacts with BMAL1 as a coactivator, but at night it can interact with PER2, which is a repressor, and perhaps might act as a co-repressor. We don't know if that's true yet. Okay, so because of these genome-wide changes in RNA polymerase II occupancy across the genome, we wanted to then additionally look at the distribution of histone modifications across the genome, which are traditionally associated with the process of transcription. And so this is a summary of different types of histone modifications that are known. In particular, histone methylation and acetylation are well-known marks that are associated with transcription and promoter regulatory regions. And so again, using ChIP-seq, this is a browser view of the PER1 gene. And to get you oriented, here's BMAL again. Then this is a activation mark called histone 3 lysine 4 monomethylation, which you can see is sort of high across the gene body. And then a classic promoter mark, histone 3 lysine 4 trimethylation, seen here, is extremely interesting. It's highly dynamic. It's not there at 0 and 4, peaks at 12, and then falls off. A second promoter activation mark, histone 3 lysine 9 acetylation, also is highly dynamic and circadian, as is the promoter and enhancer mark, histone 3 lysine 27 acetylation, shown here in orange. These two elongation marks, histone 3 lysine 36 trimethylation and histone 3 lysine 79 dimethylation, you can see are active across the gene body, but there isn't so clear circadian modulation of those marks as we see with the promoter activation marks. This is a second example at the DBP gene. Again, you see BMAL here at the top. This is the activation mark lysine 4 monomethylation, and then very striking circadian occupancy of lysine 4 trimethylation and lysine 9 acetylation, lysine 27 acetylation. So histone modifications are clearly regulated on a circadian basis. How pervasive is this kind of modification across the genome? And so to look at this more carefully, we have profiles for both RNA polymerase II and these 3 promoter marks at the transcription start site of genes across the genome. And so here at the bottom are all the 1,400 intron cycling genes. And each color of the trace represents a different time of day. You can see here. And what you can see is both polymerase marks as well as these histone marks all are dynamic and cycling throughout the day. This is especially true for lysine 4 trimethylation, shown here. So these are for the 1,400 cycling genes. This is sort of what we expect. But the surprise is if we look at all expressed genes shown at the top, about 12,000 genes, the average of these genes also exhibits cycling. Okay? If we look at the genes that are not expressed in the liver by RNA-seq, we do not see any of these marks. The reason we did this is in embryonic stem cells, it's typical for inactive genes to carry RNA polymerase marks with different histone modifications, such as K4 trimethylation and K27 trimethylation. We do not see any of this in the liver, these so-called poise genes. And then finally, these histograms at the bottom show the phase and number of genes that carry significant circadian cycling of these histone modifications across the genome. And the big surprise here is the sheer number of the genes that are cycling. So 4,000 to 5,000 genes in the liver genome are carrying histone modification sites that are cycling on a circadian basis. Much more pervasive than we ever expected. So finally, these marks are also found at enhancer sites in addition to promoter sites. And so we can look at different types of promoter sites, either defined by BMAL in this case or other factors. And what we see is that BMAL occupancies at either promoter or intergenic enhancer sites are cycling in the same way. Polymerase is cycling, of course, at the promoter, but also can cycle somewhat at enhancer sites. And then these 2 promoter activation marks shown here are cycling not only at promoters, but also cycling at enhancers. So we're seeing this kind of circadian regulation not only at promoter sites in the genome, but also at intergenic enhancer sites. And finally, the biggest surprise is here at the bottom. If we look at those genes in the liver that are expressed but not cycling at the RNA level, we see that they are still carrying polymerase marks and histone modifications that are cycling. So it's not really whether the gene is cycling or not at the RNA level that's determining whether RNA polymerase occupancy is circadian or whether histone modifications are circadian. Rather, many more genes across the entire genome are being modulated on a circadian basis, both at the level of RNA polymerase occupancy and histone methylation and acetylation. So what is really best correlated with occupancy of these factors? And so it turns out the best predictor of occupancy is whether or not the gene is expressed. So whether the factor is a circadian transcription factor, a general coactivator, or RNA polymerase II itself, there's a 90% concordance between occupancy and RNA expression from that gene. That's a very strong correlation, which of course is very low for unexpressed genes. But the surprise is that cycling genes are not especially enriched for circadian factors. Rather, the circadian factors are acting more like general transcription factors. They are directly correlated with gene expression per se, not cycling. So to try to put this all together, if we try to map out these modifications during the day on a circadian basis, what we see in the liver is an interesting set of changes that are occurring. In the morning at CT0, we see a new state that we didn't see before, and that is that clock and BMAL are bound to the DNA, yet the gene is not turned on. And we think the reason for that is that Cry1 is also bound to clock BMAL to repress that complex. The complex can still recruit polymerase II, and so we find initiated RNA Pol II with a serine 5 mark also peaking at 0. So we think that there is actually a clock BMAL Cry1 trimeric repressed complex that's recruited Pol II at 0. Then Cry1 repression dissipates, and as that occurs, we then get a wave of transcription factor occupancy, BMAL clock NPAS2. This is accompanied by P300 coactivator recruitment, and we also see K9 acetylation as well as K4 monomethylation activation marks occurring at this activation phase. This is then followed by this phase of nascent transcription, which is accompanied by polymerase II hypophosphorylated Pol II. And then shortly after that, we find the beginning of the repression phase, where the repressors all begin to occupy and bind. Clock and BMAL occupancy falls off, and then the repression phase wanes, and then we start the cycle over again. So that's an overall summary of what we think is going on at the transcriptional level in cells in the liver as far as the circadian clock is concerned. And what is really surprising in this work is that the clock is really not only controlling cycling genes, but is really controlling basic machines of the cells, such as RNA polymerase II itself. The RNA transcription machine of the cell is being recruited on a daily basis across the entire genome of the liver, and that's really a surprise. So I'd like to acknowledge all the people who are involved in these two major projects, the crystal structure of clock and the genome-wide analysis of clock BMAL and its target genes. Thank you.